You might think that only your proteins are the functional polypeptides, like we won't care about your dipeptides, tripeptides, and so on. You're wrong. Even dipeptides, they have functions. Now, the shortest, biologically, take note, biologically functional peptide is a tripeptide. So this tripeptide is called glutathione, and it's made up of glutamic acid, cysteine, and glycine. Now, this one is kind of unique because the peptide bond between the glutamic acid and cysteine residues are not formed from the alpha carboxyl group of glutamic acid. So if you look at the structure, you have here your alpha carbon. So you have there your carboxyl group, which is not part of your peptide bond. So this carbonyl in the peptide bond is actually from the carboxyl in the side chain of glutamic acid. That is why in the long name of glutathione, you have gamma glutamyl cysteine glycine. What is the function of this tripeptide? This is what we call an antioxidant. In fact, I think you have heard of glutathiones in some commercials or ads, those whitening products. So they say that their supplement has glutathione, which has this whitening effect. Actually, the main function of glutathione is an antioxidant. The whitening is just a side effect. So what does glutathione do? This cysteine side chain has a sulfhydryl group, right? The SH. If you have two glutathione molecules, the sulfur here can undergo reduction, and what you will get is an oxidized glutathione, which is shortened into GSSG. So the sulfur from each of the cysteine residues are bonded to each other. They become bonded to each other. So in the process, it releases two electrons. Now, what is this about? Undergoing oxidation. Why? So it's to protect other biomolecules like your DNA, the lipids found in your cell membranes, carbohydrates. So, for example, UV rays, they cause oxidation of your biomolecules. An example would be your DNA. UV rays can cause structural changes in your DNA. These structural changes can cause mutations, and we don't want that, right? So the body has its naturally synthesized glutathione, and the glutathione are the sacrificial molecules. So they are the ones that get oxidized instead of your biomolecules. So this oxidation of your glutathione prevent the oxidation of other biomolecules. That is why it's called an antioxidant. Now, if you have the oxidized form of glutathione, so that's GSSG, this can be recycled by the body. So we have an enzyme that makes this possible. It's called the glutathione reductase. So the oxidized glutathione gets recycled. So again, you have two glutathione molecules. These two glutathione molecules can again get oxidized, okay, in place of other biomolecules. Now, aside from this glutathione, you have other short peptides that have functions. We have here a dipeptide. So do you recognize the amino acids? So you have here aspartate and what is this? Phenylalanine, right? So the N terminal, you have aspartic acid or aspartate. And then In the C terminal, you have the phenylalanine with its carboxyl group modified. So it's an ester instead of a carboxyl group. This dipeptide is more commonly known as aspartame. Do you know what aspartame is? It's your artificial sweetener, right? See, it's just a dipeptide, but it has a function. Next, this one. This is a tripeptide. Can you recognize the amino acid residues? This is a hormone. It's called the TRH, or thyrotropin releasing hormone. So it's the hormone that causes the release of another hormone called thyrotropin. So have you recognized the amino acid residues? There's proline, right? There's proline. And then we have histidine. Do you recognize this? The imidazole group. Now, the C-terminal residue, I don't think you will be able to recognize this, but this is actually glutamic acid with the carbonyl group fused to the amine group. This is called pyroglutamic acid. And then the last example in this slide, this is a complicated peptide. It's a poison found in some species of mushroom. This is called amanitin. Okay. Other, some antibiotics actually, some antibiotics are peptides. So they also have this quite complex structure like amanitin. Now, on the other end of the spectrum, we have the largest protein. It's called titin. It's composed of 34,350 amino acids. This is I think, the recent count. When it was first discovered, they have a lower number of amino acids. Its molecular weight is 3,816 kilodaltons, or more than 3 megadaltons. And it is found in muscles. So it is a molecular spring that provides elasticity in our muscles. Now, your titin is made up of around 300 units of these globular structures. So this is a straight chain protein, and this one is just the 65th to 70 of the 300 plus units found in one titin molecule. So it's very big. And if again you notice the structure, it's elongated, right? So the elongated structure can be related to the function of titin.